Hello everyone, and welcome to today's presentation. I'm excited to have you here. Today, we'll be exploring the fascinating world of amylolytic enzymes. The presentation is part of the BTC 3101 Enzyme Technology course. Over the next few slides, we'll cover what these enzymes are, how they work, and their applications. Let's get started. So let's start by asking, what is amylase? Well, amylase is an enzyme, which means it's a special protein that helps speed up chemical reactions in the body. Its main job is to break down starches, those complex carbs we find in foods like bread, pasta, and potatoes, into simpler sugars like glucose that our bodies can absorb and use for energy. This process happens primarily in our saliva and pancreas, which highlights how important amylase is for digestion right from the moment we start eating. Understanding how amylase works is really key because it connects to the overall theme of how our body efficiently processes the food we consume. So, in essence, amylase is a crucial player in turning the food we eat into the energy and nutrients our bodies need to function properly. So, where can we actually find amylase? Well, this enzyme isn't something we have to look far for. It's naturally present in several sources, particularly in certain plants, microorganisms, and even in our own bodies. For example, amylase is abundant in saliva, which starts breaking down starch right in our mouths. That's why you'll notice that when you chew bread or pasta, the process of digestion begins almost immediately. Beyond saliva, amylase is also produced in the pancreas, which releases it into the small intestine to help digest larger amounts of starch from our diet. If we're considering industrial or commercial sources, we can extract amylase from microorganisms like fungi and bacteria. These microorganisms are cultivated specifically to produce this enzyme in large quantities for various applications, such as in brewing, baking, and even in the production of biofuels. So, understanding where amylase comes from is key because it helps us harness and utilize this enzyme effectively, whether for health purposes or industrial processes. And of course, knowing its natural sources ties back to the overall theme of how biological enzymes play essential roles in both our bodies and various industries. So let's talk about the substrate for amylase. Essentially, the substrate is what amylase acts upon. In this case, it's primarily starch. Starch is a complex carbohydrate made up of many glucose units linked together. When amylase encounters starch, it breaks down these long chains into smaller, more manageable sugar molecules like maltose and eventually glucose. This process is crucial because it's how our bodies convert the foods we eat into usable energy. Understanding the substrate helps us see how enzymes like amylase are highly specific. They only work on particular molecules. So, in the context of digestion, the substrate is the starting point for enzyme action, linking directly to the overall theme of how our bodies efficiently process nutrients. So let's take a closer look at what the substrate of amylase actually looks like. When we talk about substrates, we're referring to the molecules that enzymes like amylase act upon. In this case, the substrate is starch, which is a complex carbohydrate made up of long chains of glucose molecules. These chains can be either linear or branched, and that's important because it influences how enzymes break them down. The substrate structure provides the specific binding sites where amylase attaches. Think of it like a key fitting into a lock. The shape and chemical properties of the starch allow amylase to recognize and bind to it efficiently. Once bound, the enzyme catalyzes the breaking of the bonds between glucose units, turning the complex starch into simpler sugars like maltose. Understanding the substrate structure helps us appreciate how enzymes are so specialized. The shape and chemical makeup of the substrate dictate how well the enzyme can interact with it. This specificity is crucial because it explains why enzymes only catalyze particular reactions. So, in the big picture, recognizing what the substrate looks like helps us understand the entire process of digestion at a molecular level. It's the first step in how enzymes like amylase perform their vital role in breaking down food and providing energy. So, how does amylase actually react on the substrate? Well, amylase is an enzyme that specifically targets starch molecules. It works by binding to the starch and breaking down the long carbohydrate chains into smaller, more manageable sugars like maltose and glucose. This process is called hydrolysis, where the enzyme uses water to cleave the bonds between the sugar units. The key here is the enzyme's active site, which is perfectly shaped to fit the starch molecules. When the substrate binds to this active site, the enzyme stabilizes the transition state and lowers the activation energy needed for the reaction to occur. This makes the breakdown process much faster than it would be on its own. Importantly, this reaction is highly specific. Amylase only acts on starch and not other types of carbohydrates. And as the reaction progresses, the substrate is converted into simpler sugars, which can then be absorbed in our digestive system. Remember, this reaction is a perfect example of how enzymes speed up biological processes in a very targeted and efficient way, playing a crucial role in digestion and overall metabolism. Let's take a closer look at how amylase can be applied in the industry. Amylase plays a crucial role in various industrial processes because of its ability to break down starches efficiently. One of the primary applications is in the production of high-fructose corn syrup, where amylase helps convert starches into sugars that are used in sweeteners. Additionally, it's widely used in the brewing industry to convert starches in grains into fermentable sugars, which are essential for alcohol production. Beyond that, amylase is also used in the textile industry to remove starches from fabrics, improving their quality and appearance. Overall, these applications highlight how versatile and valuable amylase is across different sectors, connecting back to the broader theme of enzyme utilization in industry to improve efficiency and sustainability. Let's take a moment to look at the learning outcomes. These are the key points we aim to understand by the end of this section. First, we'll explore the main concepts thoroughly, but in a conversational way, making sure they're clear and relatable. I want to highlight what's most important, those core ideas that really drive the message home. And as we go along, I'll connect these points back to the overall theme of the presentation, so everything fits together seamlessly. Remember, the goal here is not just to memorize facts, but to really grasp how these ideas play a role in the bigger picture. Amylolytic enzyme, or amylase, is a fascinating group of enzymes that play a crucial role in our digestion. These enzymes are responsible for catalyzing, or speeding up, the hydrolysis, which is essentially the breakdown, of complex carbohydrates like starch and glycogen. Think of them as molecular scissors that cut these large molecules into smaller, more manageable pieces. Specifically, amylase breaks down starch and glycogen into smaller units called oligosaccharides, disaccharides such as maltose, and even into simple sugars like glucose. This process is vital because our bodies can only absorb and utilize these smaller molecules, especially glucose, for energy. Understanding amylase helps us see how our body efficiently converts the foods we eat into usable energy. It highlights the importance of enzymes in metabolic processes and how they facilitate the digestion of complex carbs, which is a key part of our overall health and energy management. Let's take a closer look at how amylase interacts with starch, which is a complex polysaccharide. Amylase targets the alpha-14 glycosidic bonds within the starch molecule, breaking down these linkages to facilitate digestion. It can also act on the alpha-16 glycosidic linkages, although primarily it focuses on the alpha-14 bonds. Now, there are different types of amylase enzymes based on how they attack the starch. Exomylase, for example, works from the reducing end of the molecule, gradually removing glucose units. In contrast, endoamylase acts internally, cleaving bonds within the polymer strand to produce shorter chains. The core process here is hydrolysis. Amylase catalyzes the breakdown of glycosidic linkages through hydrolysis, which essentially involves using water to cleave these bonds. This enzymatic activity is fundamental for converting starch into smaller, absorbable sugars, linking directly to the overall theme of digestion and nutrient absorption. Now, let's take a closer look at what happens during this reaction. A molecule of water is actually split apart, and this process is called hydrolysis, which means water splitting. When the water molecule breaks down, the hydroxide ion, O, and the hydrogen ion, H+, each attach to the exposed ends of the broken starch polymer. This is a key part of how complex molecules are broken down into simpler units. Hydrolysis isn't just limited to starch. It's a common mechanism used by enzymes throughout biology to break chemical bonds. Enzymes facilitate this process, making it easier for molecules to be broken apart efficiently. So, in essence, hydrolysis is about water acting as a helper to cleave bonds, which is crucial in digestion and many other biochemical reactions. This process highlights how vital water is in breaking down molecules and enabling our bodies to absorb nutrients effectively. Today, I want to talk about the hydrolysis of starch, which is a crucial process in how our bodies and various industries break down this complex carbohydrate. Hydrolysis essentially means using water to cleave chemical bonds, and in this case, we're targeting the bonds in starch molecules. Starch is made up of long chains of glucose units, linked together by alpha-14 and alpha-16 glycosidic bonds. When starch undergoes hydrolysis, enzymes like amylase come into play. These enzymes help break those bonds, converting starch into smaller, more manageable molecules like maltose and eventually glucose. This process is vital because it makes the stored energy in starch accessible for absorption and use by our cells. Understanding hydrolysis of starch is not just important biologically, but also industrially, for example, in producing sweeteners like glucose syrup or in brewing. It highlights how essential enzymes are in facilitating these chemical changes efficiently and selectively. So, in the context of our overall theme, recognizing how starch is broken down through hydrolysis helps us appreciate the biochemical pathways that support energy release and the practical applications that stem from this process. Before amylase can effectively work on breaking down starch, the starch granules need to undergo a process called gelatinization. This is an important step because these granules have a protective outer layer that can actually block the enzyme from reaching the starch molecules inside. Think of it like a barrier that prevents the enzyme from doing its job. Once the starch is gelatinized, that protective layer is broken down or softened, making it much easier for the amylase to access the starch molecules. With easier access, the enzyme can then efficiently break those molecules into smaller, more manageable components. This process of gelatinization before hydrolysis is crucial because it ensures that the enzymatic breakdown of starch is more effective. It ties into the overall theme of how various preparatory steps can influence the efficiency of biochemical reactions, especially in digestion or food processing. Let's talk about gelatinization of starch. When we heat starch in the presence of water, something interesting happens. The heat causes the starch granules to absorb water and swell. This process breaks down the crystalline structure inside the granules, making the starch more soluble. As the temperature increases, the viscosity of the mixture rises because the starch molecules, mainly amylose and amylopectin, are leaching out and thickening the liquid. This is a key point because gelatinization is essential in cooking and food processing. It helps create the textures we associate with sauces, puddings, and many baked goods. Understanding this process connects to the broader theme of how heat and moisture transform ingredients and influence the final product's texture and consistency. Let's take a closer look at substrates for amylolytic enzymes. These enzymes are specialized in breaking down starches, which are complex carbohydrates. The key point here is that their substrates are primarily polysaccharides, particularly starch. Starch itself is made up of two main components, amylose and amylopectin. Understanding these components helps us grasp how amylolytic enzymes work. Amylose is a mostly linear chain of glucose units, making it relatively easy for enzymes to access and break down. Amylopectin, on the other hand, is highly branched, which can make its breakdown a bit more complex, but enzymes are adapted to handle this structure as well. It's important to recognize that these substrates are abundant in nature and are the primary energy source in many diets, especially in grains and tubers. This connection highlights why understanding the substrates for amylolytic enzymes is crucial. It directly relates to digestion, nutrition, and even industrial applications like brewing and biofuel production. So, in essence, the substrates for these enzymes are mainly starches, complex carbs that these enzymes are perfectly evolved to target and convert into simpler sugars for absorption and utilization. Now, let's take a closer look at slide 16, which shows a microscopic view of potato starch within potato tuber cells. As you can see, starch plays a crucial role in plants, especially in storage organs like tubers. When we look at these cells under the microscope, we can observe the starch granules, which are the stored form of energy for the plant. These granules are quite interesting because their structure and size can vary, affecting how easily they are broken down during digestion. Highlighting the importance of starch, it's not just a storage molecule for the plant, it's also a major source of carbohydrates in our diets. Understanding how starch is stored and structured in the cells helps us appreciate how energy is stored in plants and made available to us. Throughout this presentation, we're exploring how different plant compounds, like starch, contribute to their growth and our nutrition. So, this microscopic view reminds us of the intricate, cellular-level processes that underpin the food we eat every day. Let's take a look at the sources of starch. Starch is a carbohydrate that we find in many common foods, and understanding where it comes from is key to managing our diet. The main sources include grains like wheat, rice, and corn, which are staples in many diets around the world. These grains are rich in starch, providing a significant energy source. We also find starch in tubers such as potatoes, yams, and sweet potatoes. These are particularly high in starch and are important carbohydrate sources in various cuisines. Legumes, like beans and lentils, contain smaller amounts of starch but are still valuable sources, especially because they also provide protein and fiber. By recognizing these sources, we can better understand how starch contributes to our nutrition and energy needs. It also helps us make informed choices about incorporating these foods into a balanced diet. Remember, the overall goal is to use these sources wisely within the context of a healthy, varied diet that supports our overall well-being. Starch is a polymer, which means it's a large molecule made up of many smaller units. In this case, those units are glucose molecules. The structure of starch is primarily characterized by glucose units linked together by alpha 1-4 glycosidic bonds. These bonds connect the glucose molecules in a straight chain, giving starch its characteristic structure. However, it's important to note that some of these glucose units are also linked by alpha 1-6 bonds, which create branching points within the starch molecule. These branches influence how starch is broken down and utilized in biological systems. Understanding the structure of starch helps us see how it functions as a storage form of energy in plants. Its branched architecture makes it more soluble and easier for enzymes to break down when the plant or animal needs energy. So, in the context of nutrition and biochemistry, recognizing these structural features is key to understanding how starch provides energy and how it's metabolized. So, when we look at the composition of starch, it's actually made up of two main types of polymers, amylose and amylopectin. These two components work together to give starch its unique properties. Amylose is a mostly linear molecule, which means it forms straight chains, while amylopectin is highly branched. This difference in structure influences how starch behaves, especially when it's cooked or processed. Understanding these two types helps us grasp why starch can behave differently in various applications, linking back to the broader theme of how starch functions in food and industry. Let's take a closer look at amylose. Amylose is a type of starch, and what's really interesting about it is its structure. It's made up of long, unbranched chains of glucose molecules, which means the molecules are linked together in a straight line. This linear arrangement allows amylose to pack tightly, making it less soluble in water compared to other types of starch. When heated with water, amylose tends to gel and thicken, which is why it's important in cooking and food processing. Understanding amylose helps us see how different starches behave in various applications. Its structure influences the texture of foods and how our bodies digest them, connecting back to our overall theme of how molecular structures impact functional properties. So, in essence, amylose plays a crucial role in determining the characteristics of starchy foods we consume daily. Now, let's take a closer look at amylopectin. Amylopectin is a highly branched polysaccharide, and it's a key component of starch. What makes it particularly interesting is its structure. Its branches make it more accessible to enzymes, which means it can be broken down more quickly than other types of starch. This rapid breakdown is important because it influences how quickly our bodies can access energy from foods that contain amylopectin. In the context of our overall discussion on carbohydrates, amylopectin exemplifies how the structure of a carbohydrate affects its digestion and energy release. Its branched nature contrasts with amylose, which is more linear and digests more slowly. Recognizing these differences helps us understand the varying impacts of different carbohydrate sources on blood sugar levels and energy management. So, in summary, amylopectin is a highly branched, easily digestible form of starch that plays a significant role in how carbohydrates are utilized by our bodies, linking back to our theme of understanding carbohydrate structures and their physiological effects. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at the different types of amylase. Amylases are enzymes that play a crucial role in breaking down starches, but interestingly, there are several types, each with its specific function. The main types are alpha amylase, beta amylase, and gamma amylase. Alpha amylase, for example, is found in saliva and the pancreas and starts the process of starch digestion right in the mouth. Beta amylase is primarily found in plants and helps break down starch into maltose units. Gamma amylase, on the other hand, works in the small intestine and is responsible for releasing glucose from the ends of starch molecules. Recognizing these different types highlights how specialized enzymes are, working together seamlessly to ensure efficient digestion. This diversity of amylases underscores the complexity and elegance of our biological systems, which is a key theme in understanding how our bodies process nutrients. Let's take a closer look at amylase first. It specifically cleaves alpha-14 glycosidic bonds between glucose units. An important point here is that amylase acts internally within the starch molecule, making it an endoenzyme. Unlike glucomylase, which works from the ends, amylase breaks bonds inside the chain rather than just trimming from the ends. Now, moving on to glucomylases, these are a bit more versatile. They can hydrolyze both alpha-14 and alpha-16 bonds, which means they can handle branched starch molecules more effectively. This ability allows them to convert starch all the way down to glucose. And an essential difference here is that glucomylase is an exoenzyme. That means it works from the non-reducing ends of the saccharide chains, removing glucose units one at a time. Connecting these points to the overall theme, understanding these enzyme specificities is key to optimizing how we break down starch into simple sugars, whether for industrial processes or nutritional purposes. All right, let's take a closer look at the different types of amylase. As you know, amylase is an enzyme that plays a crucial role in breaking down starches into simpler sugars. There are primarily two main types we focus on: salivary amylase and pancreatic amylase. Salivary amylase, as the name suggests, is produced in the salivary glands and begins the digestion process right in the mouth. It starts working on starches as soon as we chew, which is pretty fascinating. Then, once the food moves to the stomach and later into the small intestine, pancreatic amylase takes over. This enzyme is produced by the pancreas and continues breaking down the remaining starches into maltose and other simple sugars. Recognizing these different types helps us understand how digestion is a coordinated, multi-step process involving various organs and enzymes working together efficiently. It really highlights the complexity and elegance of our digestive system. Today, I want to talk about alpha amylase, which is classified as EC3211. This enzyme plays a crucial role in breaking down starches into simpler sugars, making it essential in both industrial applications and our digestive processes. When we think about how our bodies digest carbohydrates, alpha amylase is one of the key enzymes involved, starting the process right in our mouths with saliva. It acts on the internal bonds of starch molecules, cleaving them into smaller units like maltose and dextrins. This enzyme's efficiency and specificity make it invaluable in various industries, such as baking, brewing, and even in producing biofuels. What's particularly interesting is how this enzyme's activity can be optimized for different applications, whether it's in a controlled lab setting or within the human body. So, overall, alpha amylase is a perfect example of how enzymes are tailored to perform specific functions, and it underscores the theme of enzyme specialization that runs throughout this presentation. Let's take a closer look at alpha amylase. This enzyme plays a crucial role in breaking down starches into simpler sugars, which is essential for digestion. You'll notice it's often referenced multiple times here, which highlights its importance in carbohydrate metabolism. Understanding how alpha amylase functions helps us appreciate how our bodies efficiently process complex carbohydrates. Throughout this presentation, we've been exploring different enzymes and their roles, and alpha amylase is a perfect example of an enzyme that facilitates vital biochemical reactions in our bodies. So, keep in mind, alpha amylase is not just a name. It's a key player in converting the starches we consume into usable energy. Let's take a closer look at beta amylase, which is classified under the Enzyme Commission number EC3212. This enzyme plays a crucial role in the breakdown of starches, specifically by catalyzing the hydrolysis of alpha-1, 4-glycosidic bonds from the non-reducing ends of starch molecules. Essentially, beta amylase works by releasing maltose units, which are disaccharides composed of 2 glucose molecules. What makes beta amylase particularly interesting is its specificity. It acts from the ends of the starch chains and produces maltose rather than glucose directly. This process is vital in both biological systems, like in plant storage organs, and industrial applications, such as in brewing and the production of sweeteners. Understanding enzymes like beta amylase helps us appreciate how biological systems efficiently process complex carbohydrates, which ties into our overall theme of enzyme functionality and their importance in metabolism and industry. Let's talk about beta amylase. This enzyme plays a crucial role in the breakdown of starches. It acts on the non-reducing ends of starch molecules, releasing maltose units, which are disaccharides. This process is important in both biological systems and industrial applications, like brewing and baking. What makes beta amylase especially interesting is its specificity. It works from the ends of the starch chains, making it efficient for converting stored starch into usable sugars. Understanding enzymes like beta amylase helps us appreciate how biological systems manage energy storage and release, tying into the bigger picture of carbohydrate metabolism. Today, I want to talk about amylase, which is also known by its enzyme classification number, EC3213. This enzyme plays a crucial role in breaking down complex carbohydrates, specifically starches, into simpler sugars. It's fascinating because it acts on the alpha-14 glycosidic bonds within starch molecules, helping us digest carbohydrates efficiently. You'll find amylase in various organisms, including humans, where it is primarily produced in the salivary glands and the pancreas. Its activity is essential not only for digestion, but also for industrial applications like brewing, baking, and even in the production of sweeteners. So, understanding how amylase works and its significance really ties into the larger theme of enzyme functionality and their vital roles in both biology and industry. Let's take a closer look at gamma amylase, often written as gamma amylase. This enzyme plays a crucial role in the breakdown of starches, specifically by cleaving alpha-16 glycosidic bonds. It's particularly important in processes like fermentation and baking, where starches need to be converted into simpler sugars. Gamma amylase works in tandem with other amylases, such as alpha amylase, to ensure efficient starch digestion. Understanding its function helps us appreciate how complex carbohydrate metabolism is managed in both biological systems and industrial applications. Overall, Gamma amylase is a key player in transforming complex polysaccharides into accessible sugars. Let's take a closer look at this slide. Here, we're emphasizing the importance of understanding the key points thoroughly, but in a way that feels conversational. It's not just about remembering facts, it's about engaging with the material in a meaningful way. I want to highlight the most important concepts, because those are the ideas that truly drive our message home and connect back to the overall theme of this presentation. By doing so, we ensure that the audience not only hears the information, but also relates to it and sees its relevance. Remember, effective communication is about making complex ideas accessible and relatable, so let's keep that in mind as we move forward. Let's take a closer look at the different types of amylases listed here, starting with alpha amylase, which has the EC number 3211. Its full name is 1,4-alpha-D-glucan-glucanohydrolase, and it functions as an endoamylase. That means it breaks internal alpha-1, 4-glycosidic linkages within starch molecules, resulting in smaller oligosaccharides. Moving on to beta amylase, with the EC number 3213. Its full name is 1,4-D-glucan maltohydrolase, and it's classified as an exoamylase. Unlike alpha amylase, it works from the non-reducing ends of the starch chain, cleaving off maltose units, so it produces maltose as the major end product. Finally, we have gamma amylase, which shares the same EC number as beta amylase, 3213, but its full name is glucan 1,4-alpha-glucosidase. It's also an exoenzyme, but it can cleave both alpha-1, 4 and alpha-1, 6 linkages in glucans, primarily producing glucose. So the key takeaway here is that these enzymes differ mainly in where and how they act on starch molecules, influencing the types of sugars they produce, oligosaccharides, maltose, or glucose, which is critical for understanding their roles in digestion and industrial processes. Let's take a closer look at the table comparing the different types of amylases. First, we have alpha amylase, which carries the EC number 3211. Its full name is 1,4-alpha-D-glucan-glucanohydrolase. This enzyme is classified as an endoamylase, meaning it cleaves glycosidic bonds within the starch chain, specifically at alpha-1, 4 linkages. The major end products of alpha amylase activity are oligosaccharides, which are shorter chains of sugars. Next, we have beta amylase with the EC number 3213. Its full name is 1,4-alpha-D-glucan maltohydrolase, and it's an exoamylase. Being an exoenzyme, beta amylase cleaves from the non-reducing ends of starch molecules, also targeting alpha-1, 4-glycosidic linkages. Its primary end product is maltose, a disaccharide. Finally, gamma amylase also has the EC number 3213 and is known as glucan 1,4-alpha-glucosidase. Like beta amylase, it is an exoenzyme, but it has the unique ability to hydrolyze both alpha-1, 4 and alpha-1, 6 glycosidic bonds. This broader specificity allows it to produce glucose directly, which is a key point when considering how starch is broken down into simple sugars. To connect this to the overall theme, understanding these differences helps us appreciate how enzymes work synergistically in digestion and industrial processes to convert complex carbohydrates into simpler, usable sugars. Each enzyme plays a specific role, whether breaking bonds within the chain or from the ends, ultimately influencing the types of sugars produced during starch breakdown. Great question. So what's the difference between alpha amylase and beta amylase? Well, both are enzymes that break down starch, but they do so in different ways. Alpha amylase randomly cleaves the alpha-1, 4 glycosidic bonds along the starch chain, producing shorter polysaccharide fragments. It's like it cuts at multiple points, which helps in rapidly reducing the size of starch molecules. On the other hand, beta amylase works differently. It cleaves these alpha-1, 4 bonds from the non-reducing end, releasing maltose units one after another. So, while both act on alpha-1, 4 bonds, their mechanisms and the way they process starch differ significantly. Now you might wonder, why do both enzymes target only alpha-1, 4 bonds and not beta-1, 4 bonds? The key here is structural compatibility. The enzymes are specifically designed to recognize and act on alpha-1, 4 linkages because of the three-dimensional configuration of their active sites. Beta 1, 4 bonds are found in different polysaccharides, like cellulose, which have a different structure and are not substrates for these enzymes. This specificity is crucial because it allows the enzymes to efficiently process starch without interfering with other polysaccharides like cellulose, which require different enzymes altogether. So, understanding these differences helps us appreciate how enzymes are tailored to their specific functions within biological systems. Let's talk about the applications of amylase. Amylase is a key enzyme with a variety of important uses in different industries. One of its main roles is in the food industry, where it helps break down starches into sugars, making products like bread and beer. It's also used in the production of high-fructose corn syrup, which is a common sweetener. Beyond food, amylase is essential in the textile industry, where it helps in the processing of fabrics by removing starches from fibers. Additionally, amylase is used in the paper industry to facilitate the pulping process. In the medical field, amylase tests can help diagnose pancreatic disorders, making it a valuable diagnostic tool. Overall, the diverse applications of amylase highlight its importance across multiple sectors, connecting back to our theme of enzymatic functions and their practical uses in everyday life. Now, let's take a look at some of the key applications of amylase. Amylase plays a crucial role in various industries and biological processes. For example, in the food industry, amylase is used to break down starches into sugars, which is essential in the production of syrups, bread, and alcoholic beverages like beer and whiskey. This enzymatic activity helps improve efficiency and product quality. In the medical field, amylase levels in the blood can be measured to help diagnose pancreatic disorders or other health issues. It's a valuable biomarker for certain conditions. Additionally, in research and biotechnology, amylase is used in genetic studies and in the development of various diagnostic tools. Overall, the versatility of amylase highlights how vital enzymes are in both natural biological functions and industrial applications. It's a perfect example of how understanding enzyme activity can lead to practical solutions across multiple fields. Amylase works as a detergent by breaking down starch-based stains into smaller fragments that can be more easily removed by the detergent. The detergent formulations typically contain amylase enzymes that are activated when the detergent is added to water during the washing cycle. The amylase enzymes bind to the starch-based stains on the fabric or dishware and begin to break down the starch into smaller fragments. As the detergent agitates the fabric or dishware, the amylase enzymes work to loosen and remove the smaller carbohydrate fragments, leaving behind clean surfaces. Let's take a closer look at the slide. Here, we're emphasizing the importance of thoroughly explaining key points in a way that feels natural and conversational. It's about making complex ideas accessible and engaging for our audience. Highlighting the most important concepts ensures they stand out and are memorable. Connecting these points to the overall theme of our presentation helps create a cohesive narrative, reinforcing how each piece fits into the bigger picture. Remember, the goal is to communicate clearly and confidently, so that our audience stays engaged and leaves with a clear understanding of our message. Let's take a look at how amylase is applied in the detergent industry, using an example from Novozyme. As you can see in this video link, Novozyme has developed specialized amylases that help break down starches in laundry. This is particularly useful because starch-based stains can be quite stubborn. The enzyme works by degrading the starch molecules into simpler sugars, making it easier for the detergent to remove the stains effectively. This application highlights how enzymes like amylase are not only natural catalysts, but also essential in creating more efficient and eco-friendly cleaning products. It ties back to our overall theme of harnessing biological processes to improve industrial applications, demonstrating the versatility and value of enzymes in everyday products. Pullullanase is an endotype enzyme, which means it cleaves polysaccharides internally rather than from the ends. Specifically, it targets alpha-1, 6-glycosidic linkages, which are the branching points in molecules like amylopectin, glycogen, dextrin, and pullulan. Sometimes it's called pullulan 6-glucanohydrolase, or a debranching enzyme, because of its role in breaking those branches. An important point is that pullulanase needs a minimum chain length, at least a maltose unit, on the branching chain to be able to cleave the alpha-1, 6 linkage. So, it recognizes and acts on sufficiently long chains. Now, when we look at how amylopectin is degraded, beta-amylase comes into play. It works from the non-reducing end of the molecule in an exo fashion, releasing maltose units. However, it stops when it encounters alpha-1, 6 linkages, those branches, because it can't break them. But if we supply pullulanase along with beta-amylase, we can almost completely degrade amylopectin. That's because pullulanase removes those branches, allowing beta-amylase to continue its work. Pullulan, which is one of the substrates for pullulanase, is basically a chain of maltotriose units linked by alpha-1, 6 bonds. The enzyme hydrolyzes these alpha-glucan polysaccharides, effectively breaking down pullulan into smaller, more manageable sugars. Finally, it's classified as EC32141, and understanding its function is crucial in the broader context of carbohydrate processing and enzymatic breakdown of complex polysaccharides. Let's take a closer look at pullulanase, a key enzyme in starch breakdown. Pullulanase specifically targets pullulan, a polysaccharide composed of alpha-16 glycosidic bonds. It hydrolyzes these bonds, helping to break down complex starch structures into simpler sugars. This enzyme is essential in the production of high-glucose syrups and in brewing processes, where efficient starch conversion is crucial. Now, moving on to beta-amylase, which is another important enzyme in this pathway. Beta-amylase works by cleaving maltose units from the non-reducing ends of starch molecules. It plays a vital role in converting starch into maltose, a disaccharide that's more easily fermentable. Both of these enzymes, pullulanase and beta-amylase, are integral to optimizing starch hydrolysis. They complement each other by targeting different bonds and structures within the starch, ultimately improving efficiency and yield in various industrial applications. So, understanding how each enzyme functions helps us appreciate the complexity and precision needed in starch processing. Now, let's take a closer look at the application of pullulanase. This enzyme plays a significant role in various industrial processes, especially in starch processing. One of its key uses is in the breakdown of pullulan, a polysaccharide, into smaller, more useful sugars. But more importantly, pullulanase is widely employed to debranch starch molecules, specifically amylopectin. This debranching action enhances the efficiency of starch conversion into glucose, which is vital for producing bioethanol, high-fructose corn syrup, and other sweeteners. By cleaving alpha-16 glycosidic bonds, pullulanase helps in converting complex starch structures into simpler forms that enzymes like amylases can further process more effectively. This not only improves yield, but also reduces processing time and costs. Overall, the application of pullulanase is a perfect example of how targeted enzyme use can optimize industrial processes, connecting back to our overarching theme of enzyme innovation and efficiency. Cyclohextrin glycosyl transferase, also known as CGTase or cyclohextrin glucanotransferase, is a fascinating bacterial enzyme. It belongs to the same family as alpha amylase, specifically the glycosyl hydrolase family 13. What makes this enzyme particularly interesting is its ability to catalyze multiple reactions, but the most significant one is its role in synthesizing cyclic dextrins, or cyclohextrins, from starch, amylose, and other polysaccharides. These cyclohextrins are non-reducing cyclic molecules that have unique properties, making them valuable in various applications. CGTase is quite common across different bacterial species, especially within the Bacillus genus, including B. circulans, B. macerans, and B. stereothermophilus. Understanding CGTase's function helps us appreciate how bacteria can modify polysaccharides like starch into useful cyclic molecules. This enzyme, classified as EC24119, plays a crucial role in biotechnological processes, connecting enzymatic activity to practical applications in industry and research. Let's take a closer look at the reactions of CG-tase. This enzyme is quite fascinating because it can catalyze a variety of reactions, which makes it highly versatile in industrial applications. The key reactions include its ability to hydrolyze and transglycosylate, meaning it can break down starch molecules as well as transfer glycosyl groups to different acceptors. One of the most important aspects to highlight is how CG-tase's transglycosylation activity is crucial for producing cyclodestrins, which are valuable in pharmaceutical and food industries for their ability to form inclusion complexes. This specificity and efficiency in converting starch into cyclodestrins are what make CG-tase so valuable. Furthermore, understanding these reaction mechanisms helps us optimize conditions for industrial processes, ensuring maximum yield and efficiency. So, when we look at the broader picture, recognizing the different reactions of CG-tase underscores its significance in biotechnological applications, especially in transforming raw starch into useful products. This slide doesn't contain enough information. That's why we're moving to the next slide. Now, let's take a look at the application of CG-tase. This enzyme has a variety of important uses across different industries. One of its primary roles is in the production of cyclodestrins, which are cyclic oligosaccharides. These compounds are highly useful because they can form inclusion complexes with various molecules, enhancing solubility and stability. In the food industry, for example, CG-tase is used to improve the texture, flavor stability, and shelf life of products by incorporating cyclodestrins. It's also employed in pharmaceuticals to help with drug delivery, as cyclodestrins can increase the solubility of poorly water-soluble drugs. Beyond that, CG-tase also plays a role in environmental applications, such as breaking down starch waste in wastewater treatment. And in the cosmetic industry, it's used to stabilize fragrances and active ingredients. Overall, the versatility of CG-tase makes it a valuable enzyme for many sectors, connecting back to our theme of enzyme applications and their impact on improving processes and products across industries. Lactase, also known as beta-galactosidase, is a type of hydrolase enzyme. Its main role is to break down sugar molecules into simpler, more manageable forms. Specifically, lactase converts lactose, which is the main sugar found in milk and whey, into glucose and galactose. These two products are not only sweeter but also more soluble than lactose, which is important for digestion and absorption. The enzyme's EC number is 32123, indicating its specific classification as a hydrolase that acts on glycosidic bonds. Understanding how lactase works helps us appreciate how our bodies process dairy products, and also opens doors to applications like lactose intolerance management and dairy processing. This enzyme is a great example of how biochemical reactions are essential to nutrition and food science. Now, let's talk about the application of lactase. Lactase, as many of you know, is an enzyme that plays a crucial role in digesting lactose, which is the sugar found in milk and dairy products. Its primary application is in helping individuals who are lactose intolerant. These people lack enough of this enzyme naturally, so consuming dairy can lead to discomfort, bloating, and other digestive issues. By supplementing with lactase enzymes, either through dietary supplements or specially formulated dairy products, these individuals can enjoy dairy without the adverse effects. This application is not only important for improving quality of life, but also opens up opportunities for the dairy industry to create lactose-free products, making dairy accessible to a broader audience. Overall, the application of lactase exemplifies how understanding enzymes and their functions can directly impact health, dietary choices, and even the market. It highlights the practical, real-world importance of biochemistry in everyday life. Let's talk about invertase, also known as EC32126. This enzyme plays a crucial role in breaking down sucrose, which is a common sugar we encounter daily. Invertase hydrolyzes the terminal non-reducing end of molecules like sucrose, which is a type of alpha-fructofuranose. What this means in simpler terms is that it cleaves the bond at the end of the sugar molecule, resulting in smaller, simpler molecules, mainly fructose. The key point here is that sucrose is the primary substrate for invertase. When invertase acts on sucrose, it essentially splits it into glucose and fructose, two simple sugars that are much easier for our bodies to absorb. This enzyme is often called alpha-glucosidase or saccharase because of its specific activity. Understanding how invertase works helps us appreciate the broader picture of carbohydrate metabolism, especially how complex sugars are broken down into simpler forms that our body can utilize efficiently. This process is fundamental in many biological systems and has significant implications in food science and nutrition. Now, let's take a look at the application of invertase. Invertase plays a crucial role in various industries because of its ability to catalyze the breakdown of sucrose into glucose and fructose. This reaction is especially important in the food industry, where invertase is used to produce invert sugar, which is sweeter and more soluble than regular sucrose. You'll find it in products like candies, beverages, and baked goods to improve texture and sweetness. Additionally, invertase has applications in the pharmaceutical industry, particularly in the production of certain drugs and in biotechnological processes. Its ability to facilitate specific biochemical reactions makes it a valuable enzyme beyond just food. Overall, understanding how invertase is applied helps us appreciate its significance in both industrial processes and everyday products. Now, let's take a look at some of the key enzymes involved in converting glucose into other molecules. First, we have glucose isomerase, or GI, which plays a crucial role in converting glucose into fructose, especially in the production of high-fructose corn syrup. Then there's glucose oxidase, or GOD, an enzyme that oxidizes glucose to produce gluconic acid and hydrogen peroxide, which is important in various food and medical applications. These enzymes are part of a broader group called glucose-transforming enzymes, and their functions highlight how versatile and essential enzymes are in biochemical conversions. Recognizing these enzymes helps us understand how glucose can be efficiently transformed into different valuable compounds, connecting back to the overall theme of biochemical processes and their industrial applications. Let's take a closer look at enzymatic modification of starch and some of the key processes involved. Isomerization, for example, is a reaction where the structure of sugars is changed to produce different isomers, which can have distinct functionalities. Transfer reactions often involve moving groups between molecules, which is crucial in modifying carbohydrate structures. Hydrolysis breaks down complex sugars into simpler ones, like sucrose or glucose, making them more accessible for various applications. When we talk about sucrose and coupling sugars, we're referring to how different sugars can be linked together to form larger molecules like oligosaccharides. These oligosaccharides have unique properties, often used as prebiotics or functional ingredients. Amylose, a component of starch, can be broken down into products like maltodextrins and cyclodextrins, which are widely used in food and pharmaceutical industries. Fructose and glucose are simple sugars that are often produced through enzymatic processes. For example, high maltose syrup is made by converting starch into a syrup rich in maltose, which is valuable in brewing and confectionery. High conversion syrups aim to maximize the yield of specific sugars, improving efficiency. Ultimately, enzymatic modification of starch allows us to tailor carbohydrate properties for different applications, whether it's producing sweeter syrups or functional oligosaccharides. This process is a key part of converting raw starch into versatile ingredients for the food industry. Glucose isomerase is an enzyme that plays a crucial role in converting glucose into fructose through a process called isomerization. What this means is that it takes a 6-carbon sugar, glucose, and rearranges it to form a 5-carbon sugar, fructose. This transformation is especially important in the production of high-fructose corn syrup, or HFCS, which is derived from cornstarch. HFCS is widely used as a sweetener in many food and beverage products we consume daily. The enzyme glucose isomerase is essential because it enables this conversion efficiently, making the production process more cost-effective and scalable. Beyond HFCS, glucose isomerase is also utilized in creating other sweeteners and even in synthesizing rare sugars. These rare sugars are gaining attention because they have potential therapeutic benefits, which connects nicely to the broader theme of enzyme applications in health and industry. In short, glucose isomerase is a key enzyme that not only supports the food industry, but also opens doors to potential medical advancements through its ability to produce unique sugars. Its role underscores how vital enzymes are in bridging food technology and health innovations. All right, let's take a closer look at this slide. Here, we're emphasizing the importance of thoroughly explaining each key point, but in a way that feels conversational and accessible. The idea is to make complex concepts understandable without oversimplifying. It's crucial to highlight the most important concepts clearly, so your audience knows what to focus on. And as we connect these points to the overall theme of the presentation, it helps reinforce how everything fits together, creating a cohesive story. Remember, the goal is to engage your audience, keep it natural, professional, but also approachable. By doing so, you'll make your message memorable and impactful. Glucose oxidase is an enzyme that plays a crucial role in the oxidation of glucose. It catalyzes the process where glucose is transformed into gluconic acid and hydrogen peroxide. Now, the first key step is the binding of glucose. The glucose molecule binds to the enzyme's active site, which contains a cofactor called flavin adenine dinucleotide, or FAD. This cofactor is essential for the enzyme's activity. Next, we see the oxidation of glucose. When glucose binds, the FAD cofactor gets reduced as it accepts electrons from glucose. This results in the formation of a covalent intermediate between the enzyme and the oxidized glucose. This intermediate is a transient but vital step in the process. Following this, the covalent intermediate is hydrolyzed, meaning it's broken down with the addition of water. This releases gluconic acid, which is one of the final products, and it also regenerates the enzyme, allowing it to catalyze another reaction cycle. The last key point is about hydrogen peroxide production. The reduced FAD from the earlier step reacts with molecular oxygen. This reaction produces hydrogen peroxide, which is another important product, especially in various biotechnological applications. Importantly, this step also regenerates the oxidized form of FAD, preparing the enzyme to start the process anew. So, in summary, glucose oxidase not only helps convert glucose into useful products, but also involves a series of well-coordinated steps: binding, oxidation, intermediate formation, hydrolysis, and regeneration, that make it an efficient catalyst. This enzyme's activity underscores its significance in both biological systems and industrial processes. This slide highlights a redox, or oxidation reduction, reaction. Essentially, this means there's a transfer of electrons between molecules. One molecule, called the donor, loses electrons and becomes oxidized, while the other, the receptor, gains electrons and gets reduced. In the context of the glucose oxidase reaction, oxygen plays the role of the final electron acceptor. When oxygen receives electrons, it's reduced to hydrogen peroxide. Understanding these redox processes is crucial because they are fundamental to how enzymes like glucose oxidase function in biological systems. They help us see the flow of electrons during metabolic reactions, which is a key part of the overall theme of how these biochemical pathways work seamlessly together. Now, let's take a look at some of the key applications of glucose oxidase. This enzyme is incredibly versatile and plays a crucial role in various industries. One of its primary uses is in the food industry, where it's employed to improve the quality and shelf life of products like baked goods and dairy. For example, it helps strengthen gluten in bread, resulting in better texture and volume. Additionally, glucose oxidase is vital in biosensors, especially for blood glucose monitoring in diabetics. It facilitates accurate and rapid measurement of glucose levels, which is essential for effective disease management. Overall, These applications highlight how glucose oxidase bridges the gap between biochemistry and practical use, reinforcing its importance in health, food technology, and beyond. Tara Makasi Baniak, atas perhatian dan waktu yang tela anda berikan hari ini. Saya harap presentasi, ini dapat memberikan manfaat, dan wawasan baru bagi kita semua. Jangan raguan tuk mengunjungi situs web kami di www.upam. Edu mayantuk informasi lebih lanjut adapun pertanian yang mungkin anda miliki. Sekali lagi, terima makasih atas partisipasi anda, dan semoga kita semua bisa terus belajar dan berkembang bersama. Sampai jumpa dan semoga hari anda menyenangkan.